Welcome. Today's video is focused on custom capabilities of Eurofins DiscoverX for assay development and recombinant enzyme production. For those of you not familiar with Eurofins DiscoverX, we are a products-focused company that's based in Northern California, and we have additional sites in Missouri and Poitiers, France. And in aggregate, we have developed over 800 off-the-shelf assays using various technologies for in-house development of our over 10,000 global customers. In aggregate, we have decades of cumulative technical experience in cell line engineering and cell-based assay development, and that covers the range of early discovery through to QC lot release with bioassay development, optimization, and qualification. And in addition, we have a great deal of experience in enzyme production. Importantly, our technologies, and specifically the one that supports our catalog of over 800 assays, has been implemented successfully in global pharma, biotech, and CROs, and have been used to develop assays for screening, and over 50 billion data points have been screened, and these technologies are reported in over 2,000 peer-reviewed publications. So the technology that underpins the 800 plus assays in our catalog is called enzyme fragment complementation, or EFC. And this is based on a split beta-galactosidase reporter system shown here in this cartoon. So we have two inactive fragments of the beta-gal reporter, what we call our enzyme donor and our enzyme acceptor. And when these two fragments are separated, there is no active enzyme. But when they're brought into close proximity, either by bringing them together by fusing them to two different proteins that interact, that results in production of an active enzyme, and we can detect this by the addition of substrate. And then this is read on a simple plate reader as a chemiluminescent signal. So effectively, we've used this technology to build a diverse array of assays, and these support different target areas such as GPCRs, kinases, immune checkpoint receptors, interleukin receptors, downstream pathway readouts, nuclear receptors, etc. So it's a very robust and universal technology that can be applied to a variety of targets. And as I've already mentioned, we've used this technology to build over 800 functional MOA-based cell-based assays. So despite the diversity of the platform, it's quite possible that your target of interest is not in our catalog. And if that's the case, then we do have a custom assay development capability, which is the topic of our video today. And I'm going to break this into several components. The first is cell line engineering and assay development. We'll also cover some proof of concept and feasibility study examples that you can come to us to support, and also give you an example of our custom bioassay development, optimization, and qualification. And finally, we'll touch on the capabilities of our protein production and purification team. So our custom cell engineering capabilities are quite diverse. We have our own in-house molecular biology, vector development, and we use viral delivery systems primarily for our cell line development, either retrovirus or lentivirus. Our expertise with cell line engineering spans not just simple HEK, CHO, and UTOA cells, but diverse adherent and suspension cell models. We develop co-culture models both in culture but also working with primary immune cells as well. So depending on your needs for your assay readout, we can support more physiologically relevant approaches. For our engineered systems, we either use heterologous expression systems, such as constitutive promoters like the CMV promoter, or we have experience with inducible expressions, such as tetracycline inducible promoter systems. We also have experience using CRISPR-Cas9 for knockouts and knock-ins, which I'll talk about in a few moments. And for all of our cell lines, we perform a variety of different means to evaluate expression, depending on what's most appropriate for the target. So we can support detection of mRNA by qPCR. We use flow cytometry for cell surface receptors, or immunofluorescence to look at subcellular localization if it's required. And then, of course, we can do Westerns if that is needed. Importantly, our team has experience with many different reporter systems and functional readouts. So obviously EFC is our bread and butter. We do this, have been doing this for over 20 years. But we also have experience engineering cell lines using fluorescent proteins. We can use luciferase as well. So we develop our own transcriptional reporters using our own technology, but we can also develop assays using other luciferases. And we also have experience with evaluating secreted proteins using ELISAs. And then finally, we do perform immune function assays using our own technology to support readouts such as ADCC, CDC, and ADCP, which I will also go into later. So with CRISPR-Cas9, we are doing a number of projects right now to support evaluating modulation of endogenous expression. And we find most people are interested in using this system for knocking out specific genes to develop a clean background for functional studies, perform target validation studies, or develop cell lines for phenotypic screens. So we are doing quite a few projects to support just simple knockouts of specific genes. And as I mentioned, I'll come to this in a moment, how we do the characterization. We also are able to perform knock-ins, and this can either be disease-relevant mutations or epitope tags, and I'll give an example of that in a moment as well. And the approach that we're using is a non-viral RNP delivery system, which combines the guide RNA and Cas9 protein, and these are electroporated into the cells. So there is no selection on the cells. It's something that you can do iteratively to improve your expression. But the main benefit is that it reduces off-target editing because the Cas9 is not present constitutively in the cells. It's also an approach that is amenable to working with more difficult to transfect cell types, including primary cells. So, and one additional aspect of this is that we use flow sorting to enrich for the Cas9 transfected or electroporated cells, and this improves success rate of editing. So for all of our CRISPR-Cas9 projects, we use a variety of ways to characterize that we have achieved either knockout or knock-in. These include Sanger sequencing, next-gen sequencing, qPCR, or flow cytometry, or other functional readouts as appropriate. So just as an example of how we can use CRISPR-Cas9, we can obviously do knockouts, as I mentioned, but another approach is to actually introduce epitope tags into endogenous targets. And we've done this now for a number of targets using our own EFC technology, where we're using CRISPR-Cas9 to introduce the small enzyme donor tag into the genomic locus of interest. And as I mentioned, the use of the RNP delivery system makes this quite fast. There is no constitutive expression of Cas9 or the guide RNAs, so there's a more reduced frequency of off-target events and a higher frequency of on-target genome editing. And one of the applications for using the ED tag is for looking at proteins or drugs, I should say, that degrade the target protein, such as PROTACs that are shown here in this cartoon. And one of the advantages of using our tag for this is that the ED tag has no lysine, so it doesn't target the protein directly for proteasome-dependent degradation. It requires on targeting via the PROTAC. So This is just an example here of a graph from a cell line that we've developed in K562 cells that endogenously express BRD4, and we've tagged the amino terminus of BRD4 with our EFC tag. And you can see here that two different PROTACs that target BRD4 induce robust degradation, whereas a negative control that doesn't recruit an E3 ligase to degrade the target has minimal activity in the cell line. So this is just an example of what type of cell lines we could engineer using CRISPR-Cas9. So using more standard engineering expression systems, just some examples of typical cell engineering projects that clients come to us for are shown on this slide. GPCRs are one of the primary go-tos in our catalog, and what we find is many clients come to us for assays that hit orthologs of those specific GPCRs. So for example, a mouse, cyno, or pig assays for a particular GPCR. And we're able to do this for our arrestin readout, but also for cyclic AMP or calcium. And we also have our own unique assay format for looking at orphan GPCRs, which we call pharmacotrafficking, which traffics the orphan GPCR. It traps it in the ER, and molecules that rescue its the trafficking of the GPCR to the membrane is one way to deorphanize these GPCRs. And we found this assay format actually works really well for other target classes as well. So we also have a target engagement platform. So we do a lot of custom work for unusual or undruggable targets by other methodologies using our InCell Pulse, our InCell Hunter technology. And then probably one of the most growing regions for us is novel pathway assays. We have been using our own SH2 recruitment platform for many years and have developed dozens of assays that look at an early activation step for RTKs and immune checkpoint receptors. But we're finding a lot of interest from clients in developing downstream reporters as well. So we've been seeing an uptick in that technology using our EFC technology, but we can also develop reporters either with luciferase or GFP if required. And then finally, I've already mentioned, we do have some clients who just are interested in overexpression or knockout of specific genes that don't use a reporter. So we are able to engineer those types of assays as well. So I'm going to switch gears here and talk a little bit about an example of some proof of concept style projects that we can do to support your data generation packages. So as one example, we have a platform that we call Killer, which is an MOA reflective cell killing platform. So it allows us to characterize antibodies for clients for IND enabling studies. And most commonly, the types of projects that people are interested in are characterizing whether their IgG1 based molecules have ADCC, CDC, or ADCP activity. So some examples of data that we've generated with tool molecules are shown here. These studies are done with client molecules in existing killer cell models that we've already developed. We have roughly 40 different cancer cell models in which we're expressing a specific reporter protein, which we call killer. And these particular mechanisms result in target cell killing, which we can detect with our detection reagent. So for some clients, there may be a specific model that we don't have in our catalog, and we can develop that model as part of the project and also do the characterization of their antibodies. We perform these assays with either primary human PBMCs or NK cells for specific assay readouts such as T cell redirection, which is shown here on the right. We use primary T cells. We also have our own engineered T cells, which we call our killer CD16 effectors, which can be used for ADCC as well. And then finally, for our ADCP readout, we use primary macrophages, either M0 or M1 macrophages. So in addition to, we can do feasibility studies for any other assay readout that we have as well. But we also do a lot of custom bioassay development. So this is an area that's rapidly growing to support biologic development as the molecule moves through into phase 2 and phase 3, where a lot release assay is required. So just as an example, we do have a workflow that we use quite typically for custom bioassay development. The goal for us is to be able to take an assay that is already in our catalog and convert this to what we call assay ready, or thaw and use. And there are a number of optimization steps that we evaluate in this process. We look at culture and freezing conditions, cell density, plating time, different ligand incubation times, and so forth, all able to produce an assay-ready format that can go into, can just be thawed, and the molecule of the client can be added to the plate and run. So for those clients who also want us to do the qualification of that assay before transferring it to their CRO, we also perform those studies as well. What's involved in that is shown in these next two bullets, where we optimize the drug dilution series for the innovator drug. We establish assay suitability for relative potency. We evaluate plate uniformity and determine the replicate requirements before we perform an ICH style qualification, where we evaluate robustness, precision, accuracy, and linearity of the assay, as well as determine the stability indicating properties of the assay. So these are definitely something that we can support before you transfer it to your GMP CRO. And of course, the deliverables for these types of studies are the final qualification report. Because we are defining all of the assay conditions and actually providing the materials that would transfer to your CRO, there's an assay kit definition, and then of course the detailed assay method that we work with your CRO to make sure that they're able to run before they start phase appropriate qualification and validation. So finally, I want to touch on our protein production and purification capabilities. This started out as recombinant enzyme production services in our Poitiers site. This is a very experienced team working with enzyme production and enzyme development. They have their own in-house QC, and they work with a co-located assay development team that has a lot of expertise in enzyme assays. So this site is based in Poitiers, France, and not only do they have experience in enzyme production, but they have experience shipping sensitive biological reagents globally, because they have been doing this for decades. So they're currently using several different expression capabilities. So they have an insect cell system using the baculovirus expression system. They're able to do pilot scale expression runs to evaluate and optimize protein production prior to scale up. And this one scales quite nicely from 50 liters to 10-liter batches. They also use a HEC293 based mammalian expression system for targets where the post-translational modifications are important that they be developed in a mammalian system. And this one also, they can do up to 6-liter batch sizes. And then, of course, the workhorse is the E. coli expression systems that they've been using. They're rapid and inexpensive, and they also can be turned around very quickly, up to 20-liter batches within 1 to 2 weeks. So this, because they've been doing a lot of recombinant enzyme production over the years, they have quite a lot of experience with co-expressing multi-subunit protein complexes and with working with 50 mil pilot expressions and then scaling those up subsequently after they've optimized all of the conditions. So they have a number of purification capabilities that are available. They have multiple chromatography systems available, such as IMAC, affinity, ion exchange, HIC, and size exclusion. They generally use the approach of a rapid, inexpensive pilot scale purification first before scaling up to the full scale scale up. And they do have expertise in developing orthogonal purification schemes to generate crystal grade protein purities where required. So not many groups have those capabilities. And because they have been generating enzymes for decades, they also have expertise in enzyme treatment if it's required for certain target proteins, and providing the proteins in custom buffer formulations. Importantly, they use a number of metrics for evaluating quality, obviously a protein concentration estimate using Bradford, but also purity estimation by SDS-Page and densitometry. Where needed, they perform protein ID confirmation by mass spec and also can perform Western blots to evaluate the functional status of proteins such as phosphorylation state. So in sum, for the recombinant enzyme production services, this is an end-to-end capability for cloning expression in a variety of different expression systems, a variety of different purification options, and then, of course, they can also perform assay development using a shared Eurofins team with a lot of expertise in enzyme assay development. So in summary, we deliver you tailor-made proteins, cell lines, or assays optimized to your requirements if you engage us in our custom development capabilities. We enable you to jumpstart your drug discovery and development programs by solving difficult assay development challenges while saving you time, money, and resources. And of course, for screening, lead opt, characterization, and bioanalytical assay development, we have the capabilities of supporting all of those phases. So I'd like to thank you for your attention, and I encourage you to reach out to us at discoverx.com/CAD if you have more questions. Thank you for your attention.